I think it's a truism in science that if you can see something, you can study it. What's really exciting about GFP is it's allowed us to see so much within the cells that we were not aware of before. When I heard about GFP in that seminar in 1989, I was very excited about it. I think I was the only person in the audience that was working on a transparent animal, the nematode Centrobiditis elegans, and at the same time interested in knowing where genes were expressed. And I realized that if we could get the DNA for GFP, we could insert that into the genes we were interested in. And now those genes would cause the production of GFP, and we could see that in the living transparent animal just by shining blue light onto the animal and seeing which cells turn green. So the first experiment was to actually try to see, could GFP be made in something other than the jellyfish? At the time, most of the people working on GFP, they all thought this was going to be impossible. But the person who actually did the first experiment in the lab was Ghia.
My name is Ghia Euskerchin. I was a rotation student in Marty Chalfie's lab. When Marty called me up and presented this fluorescence project, I was actually really excited because for my master's thesis worked on fluorescence, and he thought that the project with GFP might be a good match. And in general, who doesn't like fluorescence? It's really beautiful and it's just fun to work with.
So from the very start, what I wanted and what Douglas wanted was to be able to show that GFP could actually form a fluorescent protein, one that we could shine blue light on and see green, in other organisms.
Mostly I wasn't really expecting the experiment to work. One of the big reasons being because none of the other graduate students in the lab really wanted to touch it. They kind of had looks on their faces like, yeah, good luck with that.
And so that first day in October, about a month after she started graduate school, Ghia was able to look down the microscope and actually see bacteria that were glowing green when they were stimulated by blue light.